All right, 1.5a, DNA profiling PCR. The learning intentions in this unit includes DNA can be extracted from cells. Modern techniques can be used to analyze even small amounts of DNA. And 1.5.3 says segments of DNA can be multiplied using the polymerase chain reaction, also called PCR. Um, so polymerase chain reaction is a common laboratory process in vitro. That means it's done in test tube. It's a process that makes many copies of one segment of DNA. That means it amplifies the amount of DNA. So you can start with a small copy, and by the end of the PCR process, you will end up with many copies of the same DNA. It allows scientists to do analysis on even very small segments of DNA, and this is particularly useful for forensics, gene therapy, evolutionary relationships, and gene study. The steps of PCR includes three basic steps. But first, let's learn about the ingredients. There are four ingredients needed for PCR. First, the target DNA, which is the DNA that you want to make multiple copies of. The second one is nucleotides. You need free-floating nucleotides in this process because since it is basically DNA replication, but assisted by primers and Taq polymerase, you also need floating nucleotides that can be added to become new DNA. So therefore, you have nucleotides, primers, and these are the ones that attach to the unwound DNA. So the polymerase can come along and start joining nucleotides from that point on. And then you have the Taq polymerase, which is heat resistant. Taq polymerase is not found in human. It is actually originally found in bacteria, and it is very heat resistant. The reason why we need a heat resistant polymerase is because during this process, we heat the solution up and we cool it down, and we heat the DNA up again. Now, a normal human polymerase uh, would have been denatured if it has gone through such a temperature increase and such a drop. You have denatured. But since we're using a Taq polymerase, which is heat resistant, we can perform what it is meant to be doing, even after such extreme conditions. So the first step is called denaturation, and this is when you heat up everything to 96 degrees to denature the DNA strands. So the double helical structure becomes single-stranded template. The second step is called annealing. And this is done between 55 to 65 degrees, but in textbook we just call it 55. This is where we cool the reaction so primers can bind to DNA, the single DNA strands. And the last step is denaturation. Here we heat up the solution back up to 72 degrees, and that is to raise the temperature so that Taq polymerase can extend primers, synthesizing new strands of DNA. If you start with one strand of DNA, by the end of this process, you will have two. If you have two, by the end, you will have four. And likewise, if you have eight, you have 16. The process of this rate of growing in number is exponential, because every time is timed by twice the number you had before. So after having repeat this process 30 to 35, 40 times, and that will take usually, more than 30 minutes, you will end up with a lot of DNA in a really shorter time. Cycling PCR. The cycle repeats 25 to 35 minutes, and it's about 5 minutes per cycle. And by the end, you can create billions of copies in a really short amount of time. And that is everything. Pretty simple. This lesson we just learned about PCR, polymerase chain reaction. What does it do? Well, it does just one basic thing. Makes many copies of DNA so you can use it for forensics, gene therapy, and other purposes. Well, thank you. That is 1.5a.